All right, hi there. Welcome to part 9 of FRQ practice questions. This is the last part of practice test number 3 from this year, question number 1, part D. So here's the last bit. This was the question about the eukaryotic cells that infect your intestine or digestive system, and they're treating with 2 different chemicals trying to inhibit the enzyme. So for D, the last part here, researchers are discovering a strain of C. parvum that expresses a functional variation of the lactate dehydrogenase gene. So this non-functional one, it's not that it's always non-functional, it's non-functional when the chemical was added, right? Like in the graphs we saw here, it dropped in growth. So there's another form that is functional. A DNA sequence comparison shows the variant differs in the normal sequence in the region that codes for the enzyme's allosteric site. So allosteric is the key word here for review. Remember, I would do something like this with my hand, like A is the active site separated by the double L, O is some other site on the enzyme. So if we're talking about an allosteric site, this is non-competitive inhibition with this glossy pol and FX2 chemicals because they're not bonding to the active site, getting in the way of the enzyme doing its job. They are binding to some other site and causing a change in shape of the enzyme. So you should have this picture back in your notes from a while ago. If the inhibitor's not there, active site is good to go and combine to the substrate. Lactate dehydrogenase can do its job, but if they added the chemicals in the allosteric site here, it can't do its job. But we're finding this functional form that is actually allowing it to keep functioning. So predict the effects of FX2 treatment on C. parvum cells that express this variant of lactate dehydrogenase. So we're making a prediction. That's what part D is all about. They give you some more information on top of this experiment. You kind of go one step further. So prediction here is if we have a functional variant, chemical's not going to be able to help it, right? It's not going to inhibit growth of cells. You would say that FX2 has little to no effect. That's enough of a prediction. You're not going into detail. Prediction is like, what is the outcome? Increase, decrease, stay the same, double, whatever, right? So now the follow-up, provide reasoning to support your prediction, right? When you're providing reasoning, that's when you're adding the science content or the biology information that made you come up with that prediction, okay? So that's where the enzyme review comes. Yes, move it there. Move, okay. The allosteric site is where the non-competitive inhibitor binds. That region isn't the same anymore, right? It's likely that it's changing the sequence of this region that decreases the inhibitor being able to bind. So the inhibitor may be present, but it's no longer able to inhibit, right? The enzyme can work freely in a functional form. And the last one, explain how glossy pollen FX2 might be used as drugs to treat C. parvum infections. This, again, is a cell that gets inside of our digestive system, makes us sick. We're trying to kill that cell, but not us, okay? Explain why these drugs treat C. parvum infections in humans without negatively affecting human cells. So from the text, these cells don't have functional mitochondria. This whole lactate dehydrogenase enzyme is their way to go from glycolysis to fermentation and back again. That's not how our bodies work as humans. This is another foreign organism inside of us doing this that doesn't have mitochondria. So we can treat the drugs that are working on this enzyme, because we don't have that enzyme. Not sure here in this text, right? But this chemical pathway is not our human typical chemical pathway. So you can kind of use a workaround here and not need to hurt ourselves in the process. So the drug prevents C. parvum from generating ATP. It needs to survive. So I think that's a good one because it's the key enzyme in fermentation so that you can go back and do glycolysis, okay? So you basically put the brakes on the fermentation part, so it's going to run out of electron carriers from glycolysis because they're not coming back empty. Since human cells have mitochondria, they do not rely on fermentation to produce ATP. They are not likely to be harmed. So in D, the explained one here, you can see it takes a little bit more. You're not just like stating something, you're kind of connecting why humans aren't affected, what do you know about the science, why is this a good treatment. So you got to make a little bit, this answer here is like 2 sentences to it. You don't have to write a novel, but you do have to get the right content out there to connect to your prediction. Or not the prediction, the claim of the drug. Okay, so that is the end of Practice Exam 3, FRQ number 1. I'm going to do number 4. It looks like it's a little bit more ecology based, but there is some energy stuff in there, just to give you another experience of like what a number 4 looks like, even though this content specifically is not going to be covered. So on to part 10, right? See you later.